Gene therapy for genetic disorders. In vivo and ex vivo. In vivo gene therapy means therapy that is performed inside the body. It is the process of direct insertion of functional copy of a gene to a patient's body either systemically or locally to a specific organ using a vector to treat a mutated or missing gene. It is preferred for monogenic disorders that affect one specific gene in the body. Common organ targets are eye, brain, liver, and skeletal muscle. In vivo viral vector therapies are usually confined to a one-time delivery due to the innate immune response to the virus that prevents redosing. FDA-approved in vivo gene therapies include Luxturna, which is a one-time treatment used to treat people with genetic vision loss, and it was approved in December 2017. Secondly, Zolgensma treats spinal muscular atrophy in less than 2 years old children, and was approved in May 2019. Now, let's see how in vivo gene therapy is employed for treating hemophilia. Hemophilia is a monogenic, X-linked recessive bleeding disorder, caused by factor VIII deficiency in case of hemophilia A or factor IX deficiency in hemophilia B due to genetic mutations in factors VIII and IX respectively. Factor VIII synthesis takes place in the liver sinusoidal endothelial cells, and factor IX synthesis occurs in the hepatocytes. In hemophiliacs, factor VIII or factor IX deficiency causes bleeding into joints, muscles, soft tissues, resulting in joint damage, disability, and chronic pain. Valoctocogene roxaparvovec was the first gene therapy licensed for hemophilia A treatment, and it became available in August 2022 in the European Union. Etranacogene dezaparvovec was approved as the first gene therapy for hemophilia B in February 2023. This figure illustrates an example of in vivo treatment for patient with hemophilia. A healthy copy of factor VIII or factor IX transgene is packaged into adeno-associated viral vectors. This therapeutic transgene is infused intravenously into the body. Subsequently, the AAV vectors carrying the functional genes targets the liver cells, called hepatocytes. Transgene product, which is the coagulation factor VIII or IX, is then released into the circulation to restore the blood clotting function in hemophilia patient. In vivo approach for treating blindness. Luxturna was the first gene therapy for inherited blindness, approved by FDA in 2017. Blindness is due to mutations in RPE65 gene, which encodes a retinal protein needed for normal eye vision. Viral vector carrying a healthy copy of RPE65 gene is injected directly into each eye of the patient, to improve eye vision. Ex vivo gene therapy means therapy that is performed outside the body. It is the process of removing specific cells from a patient's body, modifying them in a laboratory, and then transplanting these altered cells back into patient's body using a vector. Frequently applied to cell types like hematopoietic stem cells, B cells, and T cells. It is most often used in treating blood-related disorders, such as leukemia, lymphoma, immunological diseases, and genetic disorders that affect tissues and organs. Tecartus and Zynlonta are the FDA-approved ex vivo gene therapies to treat types of lymphomas. Ex vivo gene therapy approach for treating SCID. SCID stands for severe combined immunodeficiency, which is a rare genetic disorder caused by deficiency of adenosine deaminase enzyme that affects the immune system. SCID results from the loss of function of a single ADA gene coding for the ADA enzyme. A 4-year-old Ashanthi De Silva was the first SCID patient to receive ex vivo gene therapy. It was performed by French Anderson in 1990. In Ashanthi's case, the absence of ADA enzyme prevented her body from producing the blood cells that she needed to fight infections. This figure illustrates an example of ex vivo treatment for SCID patient. This is a bacterial plasmid with cloned functional human ADA gene, and it is incorporated into a genetically disabled retroviral vector. T cells with disabled ADA gene were collected from SCID patient and cultured. Functional copy of ADA gene was transduced into the target cells via modified retroviral vector. T cells with corrected ADA gene are selected and amplified in vitro to verify the expression of ADA gene. The transfected cells with new functioning ADA gene are re-injected into the patient body to produce ADA. As the WBCs originally isolated from Ashanthi's body, there was no risk of her immune system attacking the cells. Consequently, the WBC count had increased to normal levels within 6 months of gene therapy, and her health improved over the next 2 years. Gene therapy hadn't entirely cured Ashanthi's condition because of short-lived therapeutic effects. Ashanthi continued to receive ADA injections during the gene therapy trial period to ensure good health. Scientists were confident that SCID was an ideal target for gene therapy because of the following reasons. Therapeutic effects of SCID are reversible and do not directly cause long-term damage in the individual. The new functional ADA gene expression levels need not be tightly controlled as the ADA levels vary widely in the normal population. The gene coding for ADA is very small, which makes it easy for manipulation in vitro. The target WBCs are accessible, easy to grow, and transplant back into the patient body. There was a major breakthrough in ADA gene therapy. It was tested on a 2-year-old Salsabil in 2002, and she was the first child in the world to be completely cured of her SCID. Hematopoietic stem cells were isolated from Salsabil and reprogrammed to contain the functional working ADA gene. This therapy completely restored ADA enzyme activity in her WBCs, and she recovered even from chicken pox. Thank you for watching. Please do like and share this video. Also, click on the subscribe button and hit on the bell icon to get the notification of our latest videos.